Good morning, everyone, and welcome back to our second lecture, no, third lecture, yes, on the fundamental issues in Catholic bioethics, the program of bioethics at St. Thomas University, beginning always with a little prayer in the name of the Father, the Son, and the Holy Spirit. Come, Holy Spirit, fill the hearts of the faithful and kindle in us the fire of your divine love. Amen. Father, Son, Holy Spirit. Okay, let me dim the lights here. Hopefully that's okay for note taking. Yes. All right. So, any questions or comments from previous lectures? No. Okay. I forgot to give you like a little key of interpretation for some of my glyphs, some of my writing, which is not too understandable. And I have a few little abbreviations that I use. And I will move that slide into the first presentation because I ran across it in another course. I think it's this one. Yes. Okay. So there's just some little abbreviations that I use when you read my comments. All right. I think most people know them, but anyway, just to review. 3 dots, it means therefore. That's from way back in geometry. Remember geometry? Use the 3 little dots. Ergo, therefore. All right. Okay. Then in other words, because, versus, or against, about or approximately, and then instead of. So what I'm going to do is I'm going to capture this guy and put it here at the beginning. So you have it. All right. And as I go along, sometimes I think of others, which I will also write up. Okay. So these first 2 classes, we're still looking at somewhat of an overview of the whole program, really, how we want to take this. The first class was making that distinction from the individual origin of each one of us and the individual of the origin of the species as a whole, which addresses the issue of the supposed contradiction between creation and evolution, right? And frankly, it's not just some religious people who are falling into that apparent contradiction. Many scientists are also falling into that fallacy of putting religion in contradiction with science. But one of the main purposes of the program as a whole is to show you that far from being against each other, actually religion and science collaborate with each other to give us a fuller, bigger picture of who we are, where we're coming from, where we're going, right? Because it's respectful both of the facts that we find, the facts of nature that surround us, all right, and that we're able to detect first with our external senses, and then process and analyze with our internal senses, okay? For example, what I mean by that is that as I'm speaking right now, what is reaching your ears is vibrations. It's a sound wave that will make the eardrum vibrate, and then that is translated into an electrochemical signal through the ear nerve into the brain. And then the brain will translate that into something meaningful or not, as long as I'm speaking a language that you understand. Okay. So there's also an internal sense that interprets the information from the external senses. But that's still organic, if you will, in the sense that it resides in our brain. So respectful of the facts that surround us, and at the same time respectful of our beliefs, namely the belief in one God who has become incarnate in the person of Jesus Christ. And that makes all of the difference, because that is a central event of humanity, of human history for all times, is an incarnate God who, by doing that, he wasn't just having fun because it was no fun. He wanted to show us the way from where to where, right? Because if it's a way, then there is a transition going on from where to where. All right. And the fact that when he wants to reveal himself to us fully, he does it organically. He becomes a human being and not someone else, or doesn't stay, like, for example, an angel or something like that. No, he becomes a human being. So that says something amazing about our humanity, essentially our dignity, our human dignity, right? Okay, so picking up from all the different quarters and trying to put it together, bioethics lends itself as this exercise because it has the bio and it has the ethics. In other words, it has the organic component to it, and it also has the reasoning, the behavioral, what we should be about, which is good, proper behavior. That's what ethics is about. All right, and then I said that on the individual side, we're going to kind of bracket that until next semester when we get into the course on beginning of human life, bioethical issues at the beginning of human life. And there are many, many issues there. It's an increasing number. Again, these fields are being driven by technology a lot. And we see that technology is able to do things today that was science fiction a few years ago, right? And with technology also, one of the things that I like about this program is to encourage the students to make links and to connect the dots, as they say, connect the dots. For example, from technology to economy, all right? Technology is an economic boost, right? What modern economic system is not grounded on technology? And then from technology, from economy, then politics. Because then there are political ideas and political agendas that drive a particular technology. For example, military technology, which is amazing. Do you know that there are scientific journals that are off the shelf? They are, these journals belong to essentially to the military community, and they're classified. But they are as rigorous as the journals that we read, Nature and Science, and all the other journals that are out there by the thousands in science and technology. There's a whole family of journals that are strictly secret, that they are reserved for the military and logistics and strategy and all that. I'm kind of glad that I don't have access to those journals because I don't want to know what's going on out there. Okay. All right. So we're going to get more deeply into the process of evolution in the first half of the course, and then the second half we'll look at, we'll dovetail it into the biblical narrative and what is our beliefs as Catholics, all right? Which will make a difference with regards to some fundamentalist Protestant lines that have a different interpretation of the same scripture passage. All right, so going forward then, there are about 6 lines of evidence for evolution that merge or converge in the same basic idea, which is descent with modification. Basically, if you want to distill it down to a phrase, the whole origins of species by, someone remind me please at the break, I'll bring a copy of Origins that I have in my office, the whole thick book where he has not one reference, okay? It's all kind of intuitive. He's making arguments of intuition by observation. And we can distill all that basically to these three words, descent with modification, all right? And variation. So we have these lines of evidence: the fossil record, the morphological similarities between species, then comparative embryology, which is a fascinating field, vestigial structures that are in our bodies that no longer have a function. They are typically degenerate structures, but they point to possibly a common ancestor with other creatures that also have that structure that is still functional in them. Biogeography also, in other words, the distribution of populations throughout large masses of the crust, essentially the continents. And then the more recent in the past at least half century, with all the elucidation of the genetic code and then the translation of that into a protein sequence, the molecular evidence, all right? And the percentage of homology, of similarity that is shared among different species that have, let's say, similar proteins. You understand now with a protein sequence, we can get the DNA sequence from that protein sequence of amino acids. And in fact, we can also get the direct DNA sequence today with sequencers, DNA sequencers. Again, if you have any questions or comments at any way, just stop me and we'll address it, all right? Okay, so let's start with the fossil record then. These are things that we find out there when the archaeologists and geologists and anthropologists and biologists dig. They find these structures, which are obviously not just rock. They're mineralized bodies. It's mostly hard tissue, which would be mostly for the endoskeletal animals, the ones that have an internal skeleton. It's mostly fossilized bone, all right, mineralized bone. And for the creatures that have an exoskeleton, which is mostly made out of chitin, which is a protein that hardens, then we also have at times that can also become mineralized, fossilized, or it can be embedded in amber, which is a resin, mostly from what? Anybody know the resin?
Honey or tree sap?
Conifers, yes. Pine trees, yes. Ada, you said, I'm sorry?
Honey or tree sap?
Tree sap, yes. Tree sap, but it's mostly resin, and it's a good opportunity to make a distinction between sap and resin, even though we sometimes use it interchangeably, but it does make a difference out there, especially when it gets cold, because sap has a high water content and it tends to freeze or become extremely sluggish and basically doesn't circulate much in the winter when it's below zero or below freezing. And that's for deciduous trees, and deciduous trees are the ones that shed their leaves. In contrast to that, there is resin. And resin has very little water content, and therefore it doesn't really freeze in the winter. It keeps circulating, maybe a little more slowly, but it does keep circulating. And it is the conifers, the pine trees, that have resin, and so they are able to keep living, don't go into a latent stage, but continue to photosynthesize and metabolize in the wintertime. That's also a reason why they're called evergreens, because they keep their needles, which are also adapted for winter, particularly so that snow doesn't accumulate much on them and break them off. Okay, so resin comes from conifers, mostly from pine trees, and I make this this distinction because that is also an evidence of the large amount of pine tree forests, of coniferous forests that was around the earth at different times of the life or the history of the earth, okay? in geological history, and we'll find these cycles. We'll talk more about the cycles, the geologic cycles and the atmospheric cycles of winterization and tropicalization of the Earth, long term, over hundreds of thousands of years, sometimes millions of years, that has happened on the Earth way before we have any evidence of human remains. Okay, so bottom line, these insects or other small creatures have gotten trapped into this amber and essentially preserved. Okay. In fact, I don't know, anyone see Jurassic Park? Right, okay, very interesting movie. Of course, science fiction, but it had some interesting points that the scientists, why were these little amber preserved insects important to them? You remember in the movie? They were looking after mostly mosquitoes, because they were, the mosquitoes, the idea would be that in the abdomen of the mosquito, there would still be some blood from the creatures that they were biting, okay, which would be the dinosaurs, and there would be hemoglobin, and in the reptilian hemoglobin, the hemoglobin is still nucleated, has a nucleus. For mammals, we lose, adult hemoglobin loses its DNA, loses its nucleus, it's dished out and nucleated. But for reptiles and fish and other vertebrates, their hemoglobin still has the nucleus, which means they have the DNA, which means the genome for that species.
The red blood cells.
Yes, the red blood cells, the RBCs, yes.
So, Father, there's a new book out called Mosquito.
Oh, yes.
And it's actually at my library, waiting.
Okay.
They say the mosquito is actually the deadliest predator towards man throughout history.
Oh, yes.
It's wiped out legions of people from disease to, and really like creating an evolutionary force where people were wiped out, your rivers flood.
Yeah.
Considering all of history, the disease that has been spread by just mosquitoes.
Yeah.
I don't have all the book yet.
Right.
But that is just
Yeah.
the most deadly predator in all throughout history.
I believe it. So it's called a vector, right? The vector that transmits a parasite from one host to another. And yes, I believe it, even more than the rat for the bubonic plague. The mosquito, because you think of its versatility to be able to spread fast, relatively fast to a large region and so forth. Yeah, so anyway, back to the fossil record, what we find is these fossils, and they can be dated. We'll look at the decay of the half-life of minerals, of elements, to be able to date these fossils to a particular time range in geologic history. Okay. Also, when these fossils are found, one notices that some have similar characteristics. For example, we notice that some of these fossils, well, first of all, these are all animals. Okay, even this one, which looks like a plant, is not really a plant. It's a crinoid, I think it's called. It's from the echinoderms, the family of the starfish and the sea cucumbers, sea urchins, right? And these are also known as water lilies, okay? But these are, there are some that are similar to these that live today, but not exactly the same shape. And they are very primitive, very ancient. But we notice that some of these creatures have similar structures. For example, they have ribs, okay? And they have a spinal column that is segmented, vertebrae. So naturally, we would group those together just by what is known as the gross morphology, the GROSS, the gross morphology. Others don't have that kind of internal skeleton, but they do have an external skeleton that is also segmented or articulated. So they will be grouped also in the same category and so forth. So we find similarities between these fossils in the same way that we find similarities among different species living today, like a zebra and a horse and a giraffe and a water buffalo. They have similar shape, similar structure. And when we look at the internal structure, even more so. This doesn't occur only in animals, it also occurs in plants, so that we have fossil plant record. It's a little more skimpy, the fossil plant record, because the tissue is not as amenable for mineralizing, right? But still it can happen, mostly on the cellulose. And here we have, this is essentially the trunk of a palm. And these are different primitive plants that can be seen here. Very obviously a leaf that was mineralized, which, by the way, a fossil is not just the actual structure of the organism, the plant or the animal fossilized, mineralized, all right, the remains of that organism, but also an impression. For example, a footprint, like with that famous skeleton Lucy, of bipedal locomotion. It could be a print. In this case, this leaf most likely is a print here on the rock. There are other leaves here. I cannot tell from the photo if it's actually the leaf itself that was mineralized or if it's the print and the organic material itself deteriorated away, which was mostly cellulose. Mm-hmm. This one looks like it's the actual leaf that got mineralized, and this one also, the tissue. Okay. So either way, it could be either the actual remains of the organism or the print that is left behind by that organism. Not only are these fossils around, they've been found throughout the entire world, in some places more abundant than others, but also they are found stratified, which means that there is a chronology to these fossils, so that there are fossils, the more recent fossils, let's say, that are dated to a more recent time. They tend to occur in the upper layers of the sedimentation strata, and older fossils are found in older layers of the strata, okay, in general. Now, we have to be careful here because these strata are also dynamic over long term. Geologic strata of the crust are dynamic over long periods of time, typically measured in tenths of thousands up to millions of years, that they fracture, they overlap, they go up and sideways and slide on top of each other. So this is the general pattern that is found in most cases, but there are a few cases where the strata are overlapped so that it reverses the chronology. And we have to be aware of that. That's why geologists are experts in telling what stratum belongs to what layer chronologically, all right? But you can see that this caricature here, this diagram has attempted to display that in the sense that the more primitive organisms are drawn at the bottom, all right, in older strata, and more recent ones, basically warm-blooded. Here seems to be a skeleton of the skull of a mammoth or something like that. Warm-blooded would be, mammals would be more recent.
So an earthquake might do something like that, right?
An earthquake could do it, volcanoes, and even just what is known as continental drift and seafloor spreading. We're going to get into more details about that in the environmental course when we look at the formation of the earth and the crust and so forth. But yes, you can think of everything is really dynamic. It's just a question of the time lapse, the time proportion. Plate tectonics, exactly. And so we think that the earth is standing still right now here where we are. But in fact, for example, Europe and North America are getting away from each other, separating about 1 or 2 inches a year, which is very significant when you run that up into hundreds of thousands of years or millions of years. And now that can be detected from satellite by laser triangulation very, very precisely, okay? And for a laser beam to measure an inch is a huge amount. It's very accurate measurements. Okay, so that's plate tectonics that we'll get into in the environmental course. But the point about this is that the fossil record is stratified. So we can look at the geological evidence and geological strata. I remember a few years ago, my brother and sister-in-law, they were living in Colorado. He used to teach, we're just two brothers, and he used to teach at the University of Colorado in Boulder. And so that is beautiful out the Rocky Mountains. Ever been to the Rocky Mountains? Oh, it's beautiful. It says God's playground. Gorgeous, gorgeous. No matter what time of year you go, they have the 4 seasons. But whatever season you go, in the winter, all the winter sports. In the summer, all the hiking and camping. We used to do a bit of camping out there. And you see this geologic strata just like this, all right? In the Rocky Mountains, the road is right there. And you see these formations right up again. You can see how dark this is. This is jam-packed full of coal. And I'm sure there are quite a few fossils here that could even be seen by plain sight, stuck in that coal layer, which represents essentially, what does that coal layer represent? A fossilized forest, a rotten forest. Okay, that's how we get coal. It's a rotten forest. First, we get petroleum, and then a few million years with pressure and temperature, that forest rots into the black gunk that we call petroleum. If that continues to compress, it will become coal, right? Solidify and give off gases, which are the natural gases of the earth. And if we continue to compress coal, what do we get eventually? A diamond, exactly, which is a crystal of carbon, right? Okay, so you see all these different movements that are occurring on the surface of the earth, including the crust going down toward the mantle of the earth, getting liquefied when it comes in contact with the mantle and being recycled through convection currents that occur at the mantle of the earth, all right? Which again, we'll talk about in more detail in the summer. But here there are both the surface of the earth, of the land, and the surface of the ocean are being recycled over long periods of time, being recycled into the mantle of the earth, where they get molten down into liquid, thick liquid, and then pushed up again through upwelling at some other part at the other end, where there's, for example, the mid-Atlantic ridge. Oh, here's a ridge, all right? Upwelling of mantle, and comes out. That's the largest mountain range in the world. It's right down the middle of the Atlantic Ocean. And just for visuals, the National Geographic has these spectacular maps of the bottom of the oceans. Let's see. I always have difficulty making this bigger. I know I can do this, but then it loses resolution. Let's see. So this has changed because it used to be that this image came to the center and I was able to make it bigger. But now it goes into a different website. Okay. See, there's another one around here. This one. Let's see. Maybe. Okay, this is holding some of its resolution. Here's the bar. So you see, this is a huge mountain range that goes all the way across from the Arctic Circle to the Antarctic Circle. All right, it's kind of a S shape, right in between North America and Europe and South America and Africa. And what is happening here is this upwelling of mantle that is coming up because of convection currents underneath in the mantle of the Earth, and is literally pouring up and out mantle that cools down enough to become crust. And then that crust is pushed sideways, which is known as seafloor spreading. And it will spread to either side of the ridge, meaning it will push Europe and Africa toward the east, and it will push North and South America toward the west. And that seafloor spreading is occurring in the Atlantic. We need an equivalent phenomenon in the Pacific that is going down into the mantle of the Earth. And that is essentially what is known as the ring of fire around, it's all the Pacific Rim. Pacific Rim is called the ring of fire.
So are you saying that at the top of that Mid-Atlantic ridge, it's like open and the water either goes up or comes down?
It's not open because there are several miles of crust that has already solidified, has already cooled enough to be solid, right? But every now and then there will be a crack, and those are the famous vents, the deep ocean vents, where you will see sometimes even magma literally pour out and solidify when it touches the ocean water, which I said, down there, the average temperature is about 4 degrees centigrade above zero, so it doesn't actually freeze, okay, because of the salt content. But and you have this drastic phenomenon of magma, of lava, red hot lava coming in contact with the ocean water and solidifying and giving off a lot of gases that are very toxic gases. But then you have a whole slew of bacteria and colonies that live on that. And then other animals that feed on the bacteria all the way up to crabs and fish and so forth. And that is happening at the bottom of the ocean in the Atlantic and the Pacific. And it's a very dynamic state.
So at the Mid-Atlantic ridge is one that's really getting any, it's not getting off.
Yeah, it does. It builds up, but then at some point it spreads out also, okay? It builds up and spreads out. And what is happening on the other side, precisely in the ring of fire, is that there is downwelling of the crust, see? Because it's a huge plate. The Pacific Ocean, the bottom of the Pacific Ocean is essentially a huge plate, all right, a tectonic plate. And there is volcanic activity all along the edge here. All this, the earthquakes that are happening that we see on the news periodically. That's a very active ridge. And there, the crust buckles up and also goes at the top, at the surface, it buckles up, forming essentially the mountain range of the Rocky Mountains up here and down in South America at the Andes, all right? And that's on the top part, but on the bottom it's going down into the mantle, and as it comes in contact with that red hot mantle, that crust liquefies, and it's being recycled. But that takes hundreds of thousands, if not millions of years, but the fact is that it's happening. So the ocean floor gets replaced periodically, okay? The carpet is changed periodically every few hundred thousands of years by new crust that comes up from the mantle. Okay, so just to point out that the Earth really is in a very dynamic state, and this figure here has attempted to represent all of the different events that can happen, of course, compressed in a small region. But you can see that the buckling will form mountain ranges. When there are vents that come up of mantle that break through the surface, we call those volcanoes. Here's the upwelling. Then there are shifts also that happen that displace the strata from each other. And here are some examples. For example, this You can see obviously the stratification, how it goes in zigzag here. But then the stratification was not only zigzagged because this sedimentation didn't occur this way originally, right? The sedimentation occurred horizontally and flat, layer upon layer. Then it got buckled and zigzag, and then it got shifted vertically. So imagine all the geologic activity that happened on this. This, by the way, is called a boulder field, and marmots living here. We would run across boulder fields when we were hiking up there and camping. And anyway, these huge boulders are just strewn around, okay? All this is geologic activity that has happened. So that will move that fossil record around, but one can still follow the layers, the strata, and geologists are good at this, and following the strata where it goes up and down and so forth. Okay. Here's another one that is basically horizontal, and they've drawn some little dotted yellow lines to show us where the actual strata would be or are. And then this got cut deeply by what? Water. Got cut by water, right? Over some geologic time, and it made a canyon right through it. So of course, whatever fossils were here are gonna be displaced somewhere else, and they could have accumulated maybe at the base of the delta of this river as it cut through the canyon. So the geologists, they have to know how to interpret these layers, but there's plenty of evidence that stratification has happened and continues to happen throughout geologic time. And geologic time is typically measured in hundreds of thousands of years or millions of years, so it's long term, it's long range, okay? Just like climate, the difference between weather and climate. Weather is on a daily basis, even seasonal, year after year, but that's weather. Climate is very long term, so it's hard to tell whether we are having climate change or not. We have weather change all the time. All right, so how are these strata, how are these layers, and how are these fossils dated? Dated, right, geologically, and that is the famous carbon dating and other isotope dating. So let's look at this in a little bit of detail. This is the idea that we talked about isotopes. That's why I was pointing out the strong force and the weak force, right? So the weak force has to do with isotope decay. In other words, the neutron that is in the nucleus. And normally, the number of neutrons is the same as the number of protons. So when we look at the periodic table, say for carbon, all right, carbon has 6 protons, and normal carbon has 6 neutrons also. So the atomic number, okay, gives us the number of protons in the periodic table of each element. And when we see carbon there on the periodic table, we see a 6 on top of the C that represents carbon. That 6 is the number of protons, okay, which is a fixed number. Now, what if we had a carbon with 7 protons? It wouldn't be carbon, exactly. It would be nitrogen, all right? So the number of carbons, the number of protons,
That was tricky.
yeah. The number of protons determines the element, and that's how we got the table of elements. Now, normally the number of neutrons is more or less the same number as the number of carbons, okay? And then that becomes the mass of the element. Why? Because both protons and neutrons, they have mass, right? They have mass, they have significant mass. Is it getting warm in here? Is it warm? Is it warm? No, it's okay. Okay. That's good temperature. All right, so the protons and the neutrons, they both have significant mass, all right, in contrast to the electron that has insignificant mass for wave purposes, reacting essentially to gravity. And therefore, the number of neutrons also have to be taken into consideration when we look at the mass of the element, all right? So the normal carbon then is known as carbon 12, which is both the electron, the proton and the neutrons together, 6 plus 6. But then there are isotopes, and the isotopes are the ones that have more or less neutrons for that particular element. Of course, we cannot change the protons because then we go to a different element, right? So each element will have X number of neutrons, and the number hovers around the number of protons, but sometimes it's more, sometimes it's less. And that's the number on the top, the superscript. Okay, let me make this a little bigger so that can be seen, this little table. And, you know, it's always a challenge for the master's degree how much detail to give because of this. We know that it's a balance between breadth and depth, right? So in a bachelor's degree, typically it's about breadth and not too much depth because we don't want to lose the kids. So we give them breadth and we expose them to the different fields as broadly as possible. At a doctoral level, of course, it's all about depth, as deep as one can go, all right? And so we become super specialized in the doctoral programs. But then the master's is somewhere in between. So it's always a toss-up between the breadth and the depth, especially when the class is heterogeneous and the backgrounds are so various, as in bioethics. So I'm always a little leery when I start going more deeply into a particular area, drilling down. I don't want to lose anyone in the process. Okay, and that's one of the reasons why I record this. But I think that most of you will see it as long as I do it step by step. Okay, so talking about isotopes first and understanding what is that concept of isotope. They're all of the same species. They're all of the same element.
So lithium is number 8 on the, I can't see the periodic.
Okay.
So lithium is 8.
Exactly. So no, lithium is 3. Lithium is 3 because that's the atomic number. Is the subscript is the number of protons. Okay. So we can see there hydrogen, helium, then lithium. Hydrogen 1 proton, helium 2 protons, lithium 3 protons. So the subscript tells us the number of protons. All right. The same with krypton, for example. Krypton 36 is on the periodic table. Let's look at that right here. There's krypton number 36. So 36 protons. Okay. And then the superscript is going to be the total number of protons and neutrons. So when it goes off the double, then those are extra neutrons. For example, sticking with lithium, the normal number of neutrons would be what? 3. Plus 3 is 6. So the normal lithium, the stable, what we call stable lithium, would be 6. All right? That would be the stable isotope. But this particular isotope, lithium 8, has more or less neutrons? More. How many more?
5.
2 more. Total of 5, but 3 match with the protons, right? So it has a total of 5, but has 2 extra, if you will, from the normal number. Okay? Bottom line, this is known as lithium 8, but in that 8 is also included the 3 protons. And this one is reactive. And it has a decay half-life of 0.838 seconds. So less than 1 second. This lithium-8 degenerates, loses neutrons in less than 1 second. Okay, the half-life.
Question.
Mm-hmm.
That would be the halftime, the 50% to lose one of them, or the two to become just a typical.
So what this is measuring now is mass. Okay, so we translate now, we have to go into mass to figure out the half-life. First, I just wanted for you to understand the nomenclature, which is Li, most elements are represented by 2 letters, some are represented by 1 letter, for example, I for iodine, or U for uranium, or O for oxygen, N for nitrogen. Most elements have actually 2 letters, and many of them are words in Latin. For example, Na, natrium, which is Latin for sodium. Okay. All right. So we know the subscript is the number of protons. The superscript is the number of protons plus neutrons, and when they go above the double of protons, then those are extra neutrons. And typically those are the ones that are reactive, that are radioactive. In other words, that they decay. And you can see that the decay rate, which is known as the half-life, varies from less than a second to hundreds of millions of years. And each element is specific, all right, in its half-life. Now, let's look at the half-life. Where does this half-life come from? Also, in general, we can say also in general that the larger the atom, okay, the longer its half-life, in general, but not always. As you can see here, for example, krypton has many more protons than sodium, and yet it's much more reactive. This particular isotope is much more reactive than sodium-24, okay? Even though it has more protons. So in general, but not always. We really have to go element by element. Okay, let's look at this half-life. What is this half-life? Well, it's just what the word says, really, is the amount of time that it takes for half of that mass to decay, to become half its mass, or its weight, because the weight is mass plus gravity, right? Or times gravity. All right, so let's look at carbon, which is the one that has a half-life in the thousands of years, and that's why it's so convenient, right? It's a good average time range. And also because every organism, since organisms were all made up of organic molecules, right? Every organism will have carbon, lots of carbon, yes, okay? And since the half-life of carbon-14 in particular is relatively large, around 6,000 years, then it's a convenient standard for measuring the fossil record, for dating the fossil record. Okay, so this is how the half-life works. The decay is going to be a parabolic curve, starting with the full number of protons and neutrons, which will determine the weight. Mass times gravity just gives us the weight, but this is actually, we can interpret this as mass, really, because the weight is measured in newtons. So this is mass, straight up mass in grams, all right? This particular amount of carbon weighs 10 grams. Its half-life is the time it takes for these 10 grams to become 5 grams. In other words, to decay, to lose what?
Half its mass.
Half of its mass, the neutrons, because the protons have to stay in order for it to still be carbon, okay? And that's why the balls get smaller or fewer in number, because it has lost the neutrons, thus it has become lighter in weight. And when it reaches half its original mass, I keep saying weight, half its original mass, the time that it takes for that mass to become half, that is known as the half-life. So this is percentage here of mass. At the beginning, we start with 100%. When it becomes 5 grams from 10.5, that's 50% the mass. That's the half-life. So one half-life for each element. One half-life is 5,730 years for carbon-14. Now, what is carbon-14? Is carbon that has how many extra neutrons? Extra, 2, because normally it would be carbon-12 is the normal, the more abundant, all right? Again, we look at the periodic chart and we see that carbon has 6 protons and the normal 6 neutrons will make it 12. So the most abundant, typically the most abundant and most stable element will be the double of the proton number, and that's what it's in nature. Percentage-wise, there will be some isotopes, there will be some variations from that stable, which will have extra neutrons. In this case, this one has 2 extra neutrons and it's carbon-14. It's a smaller percentage, let's say in the fossil bone and even in our bodies, but we have some carbon-14 in us, a small amount. So that becomes the half-life, and you can see that it's specific for each element, because it all depends on the number of neutrons associated with the number of protons in that particular element. So it all begins with the number of protons that establishes the element, and then on top of that, how many more neutrons, how many extra neutrons there are, and how long it takes for them to decay. At the half mass, that becomes a half-life. And that is an actual time. It's an actual time that can be measured experimentally in the lab and determined for each element. And these are the half-lives of these particular isotopes. Hmm? Yeah.
This is very confusing.
Okay.
So regular carbon has 6 protons and 6 neutrons.
Correct.
And
that is known as carbon-12, all right?
That's known, okay, that's what I was gonna
It's
ask.
known as carbon-12.
So
Exactly.
carbon-12 is 6 plus 6.
Yes,
Carbon-14 is 6 plus 8 because it has 2 extra.
exactly. And carbon-12 is stable, non-reactive, and non-radioactive. But carbon-14 is radioactive.
Carbon-12
Is
is non-radioactive.
non-radioactive is stable.
Okay, so.
It's stable,
so
it doesn't
unstable
decay.
equals radioactive.
But 14 decays, is an isotope, but it is unstable and it decays. And then we can ask the question, well, if it decays, at what rate does it decay? At the rate of losing 50% of its mass every 5,730 years.
Okay, I'm sorry.
Okay? No, please, go ahead.
What is the significance of why is carbon-14 good for determining the age of something rather than carbon-13 or carbon-15 or?
Exactly. Well, because this one has been determined with this particular lifetime, half-life, and then we can measure the rate of carbon-14 to carbon-12 that is left in the fossil, and by that way determining how old that fossil is, according to the original rate of carbon-12 to carbon-14 in nature.
But I think what she's asking is why carbon-14? Is there access to carbon-13 or carbon?
There are, yeah, we can look at the isotopes. I think there are about 8 isotopes of carbon.
Okay.
Yeah, and then I need a definition for isotope.
Yeah.
I think that living organisms normally keep a proportion. not happen is when it starts decaying. When you're alive, you keep certain balance between the two.
A
So
proportion.
you fix carbon-14 and carbon-12 at a stable rate while you're alive. The moment you die, and then weathering and non-metabolism, that's when that decay happens.
I thought
So
that's
you
what they
can,
call the spirit.
so you can trace how much carbon-14 you have left by measuring, by analyzing that sample. And if you see that that proportion is not the expected one in a living organism, farther you go, that is the dating of the sample of the fossil. Of course, but it's very
Right.
limited and thousands of years. I think beyond that.
Yeah, it's limited to thousands of years, right? So basically, right, I don't know the exact proportion, but there is a proportion, like Reese is saying, there's a proportion of carbon isotopes in our bodies and in every living creature, all right, as we fix carbon, essentially through the metabolic process. And that becomes part of our tissue. And that is a stable rate, that is a standard rate. Then, let's see, let's say, I don't know, that we have 1% of our carbon is carbon-14, all right? And that will be the standard rate for living organisms. When we find that that rate varies with a fossil, because we know the half-life, we know how much it takes for that carbon-14 to decay over a period of time, we can figure out, we can estimate what is the age of that fossil, when that fossil was living and was fixing carbon at a normal rate. So basically, it's I confess that I don't know the details of how the proportions, the numbers, the actual numbers, okay? But the carbon-14 is a standard measure because all living creatures have carbon in it and at a particular rate.
So do all organisms decay at the same rate?
Carbon decays at the same rate.
Okay. And so
It's the carbon that decays.
As we decay or decompose, we become radioactive. That's what I mean. I don't know. That's what I'm hearing. I mean,
That's one way of looking at it.
But I mean, there's got to be
Yeah.
some there to measure, right?
Radioactivity is also emits energy, but not all radioactivity is the same.
Okay.
Carbon is specific, and there's many others I have no idea.
Yeah.
Okay.
If we were to go out here with a Geiger counter now, that radioactivity, we would find radioactivity not only from the atmosphere, we would hear the clicks, okay, at a particular rate, but also the ground has radioactivity. And going back to the Rocky Mountains, for example, there is uranium in the ground, dust. And so there is an amount of radioactivity that is there. It's called background radioactivity. So it's there, okay? And we're kind of used to it because we are born into it and live through it, all right, for thousands and thousands of years. But there's radioactivity as background happening all the time. It's very small, very slight, and it's not sufficient generally to have a massive effect like the ultraviolet radiation has on us, especially on the skin. But it's there, it's the background. All right, so you can see, for example, here are the isotopes of carbon, all right? And 14 has been chosen. Frankly, I don't know exactly why, but I know that it is the one that has been standardized as a measure for measuring fossils in the range of thousands of years. Okay, what happens when we do this analysis is that these layers, oops, got too big here. These layers can be dated because of the amount of radioactivity that is left in the fossil, whether it's plant or animal. And also the strata can be dated also. The mineral experts, geologists know how to date these strata. And that's what we find. We find that the more primitive organisms, the more primitive fossils are found in the deeper layers and not vice versa. And we don't find them all in the same layer, for example, and no fossils above or below. Over time, what happens is these cladograms are built, or these branching diagrams. Each one of the branches here would be a nodule or a node, okay? And these nodes, the nodes, what are known as a common ancestor, In other words, this node here is a common ancestor between the sharks and all of the other vertebrates that we know today. Whereas this node here is a common ancestor between the Ostichthes, the bony fish, and all of the other vertebrates, but not the sharks. So there's a divergence that occurred between the cartilaginous fish and the bony fish early on. We continue up the cladogram. The next node will be the break between the fish in general and the tetrapods. In other words, the animals that developed 4 legs, okay, functional. And this node here, this common ancestor, is what we consider the original tetrapod, if you will, from which all other tetrapods developed. We continue the branching, and there could be nodes also internally, not just at the main trunk, if you will, but further branches. This is simplified. There will be more branches as we get closer to modern time. The branching will continue here as characteristics are more distinct, right? For example, here is a branch of the reptiles in general. At some point, this has to break between the legged reptiles and the non-legged reptiles and so forth. I was talking about common ancestor. Here's some actual fossil record evidence. This famous one, I'm sure you've heard about it before, the Archaeopteryx. Archaeo is a reference to primitive. Pteryx is a reference to the wings, to the primitive winged one, which has been dated to about 150 million years ago. Here's an actual fossil record of it. It's very evident that this creature is a tetrapod, is a vertebrate, has segmented spinal cord, including a tail, but it's also winged, all right? And the characteristics of the head with what are known as homodonts. We have a dentist in the room, so I'm not going to pretend to talk about teeth, but homodonts are the ones that have the same shape, right? They have all the same shape, which points toward a type of diet that was pretty specific, for which those teeth were sufficient. In contrast to heterodonts, which we have different shaped teeth for different functions, right? For grinding, for cutting, for tearing, etc. So again, from the fossil record, this is obviously a caricature of what this animal would look like based on the skeleton and building from the skeleton out. But what is totally, I don't want to say fictional, but totally unknown is the color pattern, okay? So this is an estimation what the color pattern would be. According to the structure, if this animal would be somewhat camouflaged, and looking at contemporary animals or contemporary birds, how they would have some kind of a camouflaged pattern, but that is unknown because that's totally at the surface, at the level of phenotype, and we don't have the phenotype, the external phenotype in the fossil record, right? But everything else is considered pretty true. The fact that it is a tetrapod and that it is winged with a long tail that also had feathers on it, as you can see. You know, the feather, you can look at the feather and you can say that it's essentially a very sophisticated hair. It's the same material, keratin protein, and it's extremely sophisticated hair that now allows this animal to go airborne. Another example that has been studied extensively is the evolution of the horse from what is known as Eohippus, which now has changed name into Hyacotherm. Used to be known normally as Eohippus. True horse, had the size of a dog, more or less, had the size of a dog originally, and the 4 digits were more or less the same size. But over millions of years, and again, this is in the fossil record, the middle finger became more and more prominent to the point that it is totally prominent in modern horses, and not only the middle digit, but the actual nail is the one that is bearing the weight of the animal. And that's why they're called ungulates, because the nail in Latin is unguia or ungna, and they literally walk on their nails, okay? The nail of the middle finger. And that we call today the hoof. That's what gets, we put the herradura, what do you call the herradura in English? The horseshoe. So that the nail doesn't crack, but they're literally walking on their nails. And that has been an incredible development of that middle finger to bear all the weight over about 55 million years. Here's a time scale in geologic time, which is in millions of years. It makes sense because the bulk of the mammals really flourished with that expansive radiation after the demise of the dinosaurs, because the dinosaurs, many of them were carnivores that were so ferocious they didn't allow mammals to really flourish because they would get eaten up. All right, so the only mammals that were gonna survive at that time had to be very small and tiny and able to scurry away from the dinosaurs. So this kind of matches the expansive radiation of what's considered mammals in general from about 65 to 60 million years ago. Again, based on that fossil record, now a whole cladogram can be built for all the horse-like ungulates. Here's the actual horse down here. Here's the zebra, the donkey, and this is some kind of a wild donkey that also exists. I think it's in Africa mostly, Africa and South America. All these numbers are pointing to the nodes of divergence. So we see that the closest relative to the horse today is this Przewalski horse. And the most distant would be these onager, which I think are from Africa. The E is for the genus, Equus. And then it goes into species and subspecies. But the importance about this cladogram is that this is what can be reconstructed from the fossil record and looking at the similarities, comparing the morphology, the gross morphology of the animals. Many branches die off Okay, and typically it will be just one or two species representing the divergence that had occurred over geologic time. But notice that many of the branches eventually become dead ends. When we look at the fossil record and we start putting together from all the different species where fossils have been found of animals and plants, we can start looking at the fact that animals with similarities, again, looking at the gross morphology, still stayed at the gross morphology of the external structure, all right? It matches what was the Linnean classification, starting from the largest grouping, which is the domain, and drilling in kingdom, phylum, class, order, family, genus, and species. So the animals and the plants are being classified, if you will, or grouped according to their similarities, which was a classical way of classifying the species. I want to talk a little bit more about the morphological similarities, what is known as structural homology. Let's say we look at vertebrates today. Let's concentrate on the vertebrates. At the forelimb, the forelimb is the forward limb. Okay, all vertebrates are tetrapods, meaning they have 4 legs. And of the 4 legs, 2 are in the front and 2 are in the back, toward the front of the body, toward the back of the body. So we talk about 4 legs and hind legs, or 4 limbs, F-O-R-E, meaning forward, and hind limbs, all right? Essentially the legs. And when we do a comparative anatomy of the bone structure of 4 different groups of mammals, the human, the cat, which is carnivore, the whales, which are cetaceans, right? They are marine mammals, and the bat, which is the only group of truly flying mammals, not gliding mammals, like there are gliding squirrels and so forth, but and some gliding lemurs, but these are truly flying animals that the 4 limbs have adapted fully into flight. We see that the bone structure is consistent internally, even though the function is very different, okay? For example, for the human, the forearm is used for grabbing, not for walking or swimming or flying. Unaided, right? Unaided is used for grasping. And so we have a forearm, and we have the second part of the arm. Then we have the wrist, and finally the digits, the phalanges. So all of these bones, which are named here, also carry over. The same bones exist in the other mammals, even though that limb is used for a different function. All right, so in the cat, we know, of course, is for crawling around, right? And so we see that mostly the wrist is the one that changes into an L-shaped position for grounding the animal. And the whale is very drastic because this, what we call the flipper, is actually the forelimb of the whale, is used for swimming and actually for gliding through the water. The actual swimming mechanism is going to be mostly the tail of the whale, which is horizontal, by the way, in contrast to the fish, true fish, which have vertical tails, right? But marine mammals, dolphin, whales, dugongs, etc., manatees, they have horizontal tails instead of vertical. But the forelimb, you can see that the digits are there, the remnants of a wrist, and even the humerus, the radius, and the ulna are also there. But degenerate in the sense that they no longer have the function that they had on the original tetrapods, where their ancestors were land walking animals. It is very evident that a whale or a dolphin is not able to walk around on land, okay, any longer with the super adapted fins that they have. But internally, the structures are all there, okay? So if there was no relation whatsoever between these mammals, why that kind of intrinsic structure? You would think it's a total waste and insignificant. A fin doesn't have to have this kind of structure for it to be a fin. For the bat also, you can see it's now the digits that are super adapted to be able to maintain a membrane of skin in between them in order for that wing to actually catch wind and so forth and be able to hold the animal airborne, lifted airborne, exactly, okay? And even further adaptation on the first two digits to maintain their claws so as to grip and hang when they're not gliding or flying. So we see adaptation, but adaptation's on a theme. The theme is basically the forearm. In other words, the bones are the same bones, if you will, the same groups of bones for all of the different species, even though they have very different function. So that is pointing obviously to some kind of adaptation, okay? If these animals were just created individually like that, with no relation to each other, why have this internal similarity? There's no reason for it. Okay? And in fact, these structures are not actually the best structures for the type of function that they're doing. For example, this flipper probably would be much more functional if he were more hydrodynamic, flatter, okay, but the bones make it bulkier. So if this animal was going to be designed and created ex nihilo as a better swimmer, that forward flipper would be much narrower and not have the bulkiness that it has, or even the size that it has. But it has that size and that bulkiness precisely because of the internal structure, the remnants of the bones that are left there from their ancestors being tetrapods.
With also theoretically, it could still evolve in X number of years in the future to be more.
Yes, in fact, yeah, when we extrapolate this to all of the species that are around, rather, we can say that every individual species that exists today, its destiny is to become extinct, all right, and some of its variants to eventually evolve into new species. So that's really the bigger picture. But of course, the factor here is that we are in the middle world. The middle world for us has really a maximum of about 100 years, all right? Who's gonna live 100 years? But we're talking about geologic time, which is minimally hundreds of thousands of years and not millions of years. So we don't see it in our lifetime, except for bacteria, where we are actually seeing evolution happening with the superbugs. Okay, so this internal morphology and the fact that there is such similarity and at the same time an adaptation, variations on a theme, is all pointing to some kind of common ancestor. It's the most logical explanation for this. And then in fact, when we look at the fossil record, we are finding those common ancestors. Now the fossil record is also scant. We say there's a paucity of fossil record simply because the vast majority of creatures, when we die, eventually we decompose and not much is left of us, all right? Or any of the other creatures. But what has been found makes sense. It fits into this plan of evolution, of divergence, of variation that may eventually lead to what is a totally new species. another line of evidence from the comparative embryology. All right. When we look at embryos and we see that they're most diverged, again, we're still with the gross morphology, all right? We're looking at the anatomy and mostly the external anatomy, but also internal anatomy of the organisms. We are most diverged in gross morphology at birth or at hatching, okay, once we come out of the embryonic stage or the fetal stage. We're most diverged. And that is illustrated here. For example, a newborn child, human, and a newborn fish or a newly hatched fish look very different, relatively different, at least externally. Let's stay with the external anatomy for now. Never mind that we all have a spinal cord and bilateral symmetry and so forth. But externally, we're most diverged once we're born, and then from then forward during our lifetime. The most drastic one, if we're looking at vertebrates between fish and mammals, between fish and human. But then when we go into the embryonic stage, we notice that the closer we get to the early embryo, the more similar we are. Again, in the external anatomy, the more similar we are, okay? To the point that earlier on, we find structures that are very similar to each other between mammals and vertebrates and fish and amphibians. But in mammals, particularly, we lose the function of those structures, okay? And I want to point specifically at the gill slits and also the tail, but it's very well known that even the human has a tail early on in embryonic development. And what's the reason for that tail? There is no reason. It's called vestigial. In other words, it's a remnant, it's left over from ancestors who had a functional tail. The gill slits also. The gill slits are very interesting because these are slits on the neck, literally, okay, that go across from the fish, through the amphibians, through the reptiles, birds, and mammals. We all have them early on in the embryonic stage. And then for mammals, for fish, they continue development into gills, and also for amphibians during their watery stage. Okay, their juvenile stage in amphibians, think of tadpoles, for example, before they become adult, they have gills, right? Functional gills. But in birds and mammals, that function is lost. And a secondary function has come in, which is going to make actually the bones of the jaw and some of the cartilage that makes the vocal cords, all right, and the trachea. That is a secondary function of those gill slits, but originally, in their original design as slits, they are for gills. I think I have a diagram here. Move down a little bit. Yeah, here we go. Okay, so this is, again, on the gross anatomy, on the external anatomy of the embryo for fish and for reptiles and amphibians also. They have the gill slits. We have them, birds, mammals. But then in fish and amphibians, they actually develop into true gills, as we can see here, right? Because birds and mammals, we don't breathe by gills, but we breathe by lungs. We have no longer any use for those. So a secondary use that has been traced is to develop different parts of the vocal cords, the lower jaw, and even some of the bones of the ear. It's amazing. They are derivatives of this original tissue in the embryo. secondary adaptations. For the tail is different because the tail in fish and reptiles and even birds continues functional. But in some mammals also functional. But for the human, that little tail is then reabsorbed back into the body, and the only thing that we have left is the coccyx, right? The tailbone. And we don't know we have it until we fall on it. And they are fused vertebrae. They are caudal vertebrae that are fused. And I think it's about 3 or 4 of them. Why have that structure down there, which makes no sense, right? And no function. It's a vestigial structure from pointing to common ancestors. Apparently the tonsils also, right? From when our ancestors were ruminants, they had to chew the cud and grind it because they were herbivores, meaning they were eating mostly grass. And we don't really have cellulase to digest, we mammals don't have cellulase to digest the cellulose. And so that digestion had to be done by bacteria, which we call the intestinal flora, which we now know is actually a fauna. It's more closer to a fauna than a flora. But those bacteria digest the cellulose, and then we get the product of the ruminants get the product of it, and they had several chambered stomachs, right? And it is thought that the appendix is a remnant of one of those chambers of the stomach that is still functional in the ruminants, in the cows, cattle that eat grass, right? And they have the several chambers of the stomach so that digestion can take place, that essentially a fermentation by the bacteria which are digesting the cellulose. Now, there has been found some secondary function of the appendix, right? Anyone know? Apparently it has something to do with the immune system. Yeah, with the immune system, it boosts the immune system, all right? But it's secondary to its original function. To the point that if that organ is taken out, the person can live a normal life, all right? My brother all of a sudden, about a month ago, started developing a pain on the side. Thank God his wife took him to the hospital right away because he was having appendicitis. And within a few hours, that appendix was out, okay? So that's the way it goes. That organ is known as a vestigial structure. For the whale also, we find the rudiments of a pelvic girdle, all right? And what is a whale doing with a pelvic girdle? Because that pelvic girdle is for what? Is for supporting the hind legs, okay? The hip. And a whale doesn't need hip because the whale is not walking, swimming. Whereas the forelimbs, you see, have developed super adapted to its function of gliding through the water. But the hind legs are lost. Okay, so this is what we call vestigial structures. Why these vestigial structures? It's just whim for putting them there with no function. No, they point, a logical explanation is that they point to a common ancestors, and when we trace the fossil record, which little by little is becoming more and more complete, we find that these structures do become functional in ancestors. Okay, I think that at this point it will be a good time to break because we have about the same amount of slides for the second half. So when we go to the list again, so far we've seen the fossil record that is chronological in the strata, geologic strata of the crust, the Earth crust. We have seen these morphological similarities among animals, organisms. Why would there be such a similarity if it were not that they're deriving from common ancestors, from a common, what we call a common body plan, a common body plan. That again is corroborated, is verified when we compare embryos and how those embryos are most similar the younger they are in their stage of development. I mean, you can take it to the ultimate argument. What is the youngest embryonic stage? It's the zygote, right? The fertilized egg. And all fertilized eggs essentially look the same as a sphere. It's an egg that has been fertilized, therefore now a zygote. And all zygotes look essentially the same, at least in the gross morphology, which is a sphere, microscopic. And the only thing that's going to vary is the size. So that, for example, an elephant zygote is about 10 times larger than a human zygote. Yeah, but a size, nonetheless, you can say, I've heard that you can actually see a cow egg, you know, a little tiny dot. It's about 5 to 10 times larger than a human egg. Mm-hmm. That's, other than the size, the gross morphology is essentially the same.
Genetically different.
Yes, because now you're talking about genotype, not phenotype.
All right,
Okay?
so it's indistinguishable or most similar when it's in, but just talking.
In the early embryonic stage, and then there is divergence.
Phenological.
Yes, yes. Mm-hmm. The phenotype, exactly. Phenotypically, we would say. Phenotypically. Mm-hmm. Yeah, so all this so far is based on gross morphology, right? And the beauty of it is, I'll show you in the second part of the class, is that when we look at the genotype, it corroborates, it matches the same story basically, that there's divergence in the genotype, all right? But I'm getting ahead of myself. Okay, so all the way down to vestigial structures, and really no explanation for them unless we put them in the scheme, if you will, in this theory of evolution from common ancestors, then they make all the sense in the world. There's an explanation for why these vestigial structures are still hanging around. And why some organisms are now born without these vestigial structures, because for that individual organism, that structure was eliminated and their organism is viable. I understand that some people now are born without an appendix. Yeah, small percentage of the population actually doesn't have an appendix, and it doesn't, and it doesn't affect them. Essentially, they can. Yeah. Okay.
Yeah,
So you
that's
see?
just like, you know, like X-ray, so there's no sign of any wisdom
Really?
wearing her head.
Yeah.
You know, so.
A mutant. Yeah. And that actually has an advantage, right? It has definitely a physical advantage. And I had my two wisdoms, they were totally horizontal. It's amazing how those things are just totally horizontal in there. And one started coming out and we got a cavity because there's no way of cleaning it, right? And yeah, so vestigial structures, right? Okay, let's break here and have some of that nice bread that was offered. And we'll pick up in about 10 or 15 minutes, okay? Pause recording. Here we go. Okay, so welcome back to the second half. And hopefully this will get recorded also. See the little dot there, that's what I look for, that little dot. We have been talking about the gross morphology of animals and plants as we see them today and as we see them in the fossil record. And we notice that there are similarities that is occurring among these different organisms. Though they belong to different species, they look similar to each other, all right? So that's already an intuition that there was some body plan, that was a common body plan from which all these various organisms are deriving in their similarity. We have another line of evidence for that, which is known as biogeography. Biogeography is looking at the animals and plants and also fungi that live throughout the world that have some similarity, even though they live in very different parts of the world, including different continents that are totally separated by oceans. And so again, how is it that these animals are so similar to each other, yet they are different species in their own right? And I'm using the biological definition of Meyer as we looked at it. So it's another line that points to the common ancestry of these animals with the record, the fossil record, in fact, revealing that throughout the different regions. So we can take example, the camelids, which are thought to have started in this region of North America, more or less in the Rockies next to the Great Plains, and have diverged either north or south. And as they have diverged north or south, they have also evolved into different species, but have maintained the same basic body plan. So that when we put these animals next to each other, we see that they have the same basic body shape, if you will, but are different species so that they don't interbreed, all right? The dromedary, which is the one with the one hump, and the actual camel with the two humps, which, by the way, I understand that doesn't have water inside, okay? That's the myth, but it's kind of a bony structure to dissipate heat. And the llama, the dromedary being mostly in Africa today, the camel in Asia, and the llama in South America. 3 different regions of the world, very similar species, but different species, so they don't interbreed, but the same basic body plan. If we were to look at the skeleton of these 3, it would be very similar skeleton. Then the llama has also diverged in South America to other relatives, the alpaca, the vicuña, the guanaco, right? And so we see that they have the same basic body plan, but are actually different species, right? All pointing to having some kind of common ancestor from which they diverged. So that's the one on bioregions. This leads into this topic of continental drift that I was talking about earlier. We'll look at it in more detail again in the environmental course, but for now, this is a chronology of the shifting of the continental plates over millions of years. If we go back about 200 million years, a little over 200 million years, we find one large continent, also known as a supercontinent, known as Pangaea. And Pangaea is a reference to all Earth, if you will. Pan is a reference to all. Pangaea or Gaia is a reference to Earth. That's where we get the word geo also, right? And so all of Earth, in other words, the crust, essentially the crust was all together into one large supercontinent that cracked, and the plates started diverging. The first big crack was more or less north-south. So it creates these 2 other still very large continents, one known as Laurasia and the other one Gondwana. Sometimes you see Gondwana land as a variation of it, all right? That happened about 150 million, within 150 million years. So you can see that the scale here is in millions of years. But this is really truly what we talk about continental drift and plate tectonics, Why is this crust shifting around, and why is it moving, and why is it cracking? Well, because we have to understand that the crust is sitting on top of mantle, which is molten rock, lava, which every now and then comes out when there's a volcanic eruption, all right? And it's molten, so it's liquid, thick, but liquid. So the crust is literally floating on top like a raft. And there's a shift, very slow, but real shift that is going on with the crust. And so this fractioning of the crust that has happened gradually is what is known as continental drift. Within 100 million years ago, basically we had the rudiments of the continents as we know them today. North America, a distinct plate from South America because North America broke off from Laurasia, okay, which was Europe and Asia together. To this day, Europe and Asia, even though they're considered two separate continents, are still one plate. South America rather broke off from Africa, and we can see that the eastern tip of South America fits rather nicely into the western concavity of Africa. And then India is, I find, one of the most fascinating, because India, Antarctica, and Australia were once essentially one large plate that cracked and started migrating. First, to remember that Antarctica, in contrast with the North Pole, is an actual continent, all right? But not on the North Pole, not the North Pole, but yes, the South Pole is a continent. India broke off from there and started migrating north, crossed the equator, and crashed into Asia. And essentially, it continues to push into Asia, all right? And that caused the buckling of the Himalayas, which is the largest mountain range in the world to this day. So India is considered a subcontinent, in part because of the size, and the other part because it is now fused into the southern part of Asia. Australia also became independent from Antarctica and migrated, but not as far as India, so it remains still in the southern hemisphere, and further break off New Zealand and so forth, and the Polynesian islands that are there strewn around. But you can see that if we go back again to 225 million years ago, is fairly recent in geologic time from the total time of the Earth, which we said was about 4.5 billion years, right? So 4.5 thousand million years. So this 225 million only fits within the last billion years. So it's a fairly recent phenomenon.
How did they get those numbers? Approximate?
It is obviously an approximation, but it's from geological strata, from the evidence of the mineral content. One of the oldest one, I think, is called zirconium, which is one of the oldest elements and hardest elements in the world. All right, and it goes.
We're using zirconium now for grounds.
Sorry.
We're using zirconium for grounds.
Yes, I heard, because I went to the dentist. I broke a molar the other day. I cracked a molar, and I went to the dentist. And she said, "Well, we're going to make your new crown with zirconium, so you can live for another 4 billion years." Talk about the fossil record, right? Everything else will be gone, and the crown. That's why I say the tombs of, anthropologists in the future are going to have a lot of fun in our cemeteries, because what are they going to find? If you fast forward thousands of years, they're going to find mostly metal, zirconium, and silicone in the tombs. The organic material will be gone. Yeah. Okay, so that's the geologic measurement that is done, the geologic dating. Then, so from this but I wanted to point out that 225 million years ago, certainly there were already plants and animals around, okay? And the fossil record is there. So when these continents were splitting up, there were already animals and plants in these different geographical regions, and they went wherever the raft went. So that created a geological isolation for them, so that it would favor divergence, because they no longer have physical access to the other group, to the other population of that species. Okay. So this continental drift certainly favors diversity, favors adaptation, because the continents are, as they're shifting, also keep in mind they're going into different climates, different climate zones of the world. All right. That's where we get the bioregions, of the world, which are about a dozen, more or less, depending on how they're classified. But you can see that they follow a general pattern couple of things to notice. This blue line, this horizontal line represents the equator. So first, various things to notice that is so obvious that a lot of people miss it. The bulk of the land mass of the world today is in the northern hemisphere, the bulk of it, okay? Now, one can say, yes, so we can see here, only the southern tip of Africa, the bulk of Africa really is in the northern hemisphere. You know, at least I tend to associate Africa with the southern hemisphere, but not at all. It's only from Equatorial Africa south, all right? And here's, by the way, one of the two big lungs of the world, the Congo Basin, and the other one is the Amazon, precisely the Amazon Basin. Equatorial. So this is what is known as the tropical rainforest region. Tropical rainforest. Also Polynesian Islands and India, Southeast Asia. This is where we find the largest biodiversity, by the way, in the tropical regions of plants and animals. Then Australia and most of South America. Of course, down here is Antarctica, but Antarctica is inhospitable to any plants and to most animals, except for penguins, seabirds, and some other mammals that get there. So basically what we have to do is we have to move north and find more variety of bioregions. We go into the temperate region, and you can see that more or less it has the same type of vegetation, whether it's Africa, Asia, or North America. And then parts of North America, a little more to the northern part, will have similarities with Europe. Okay. This one, by the way, this is called chaparral, the orange. I know they have it, yeah, they have it. Chaparral, which is kind of unique to the Mediterranean basin, but there's also a little bit of chaparral along the California coast because climatologically it's about the same. It's on the dry side and it's on in the temperate region of the world, and also is stratified, typically stratified with cliffs and so forth. Then we get into the northern region, the northern territories of Canada and Siberia. And that's the coniferous rainforest, also known as the taiga. Taiga is the coniferous rainforest. And finally, the Arctic region up here, all right? But you can see how generally they follow a horizontal pattern, because since these are bioregions, which is mostly vegetation, they're going to be adapting to the climate of the place, of the region, the zone, the climate of the zone. So you have a tropical zone, generally, you have a temperate zone, and then you have a northern zone or Arctic zone.
Father, I'm sorry, could you repeat where you said the most diversity is down in tropical or subtropical?
Tropical regions, tropical regions, yeah. Technically, we are in a transition between tropical and temperate. South Florida, that little green dot there, and that is known as subtropical, all right? Which by the latitude, we should be temperate, but because we get a breeze from the southeast, generally a sea breeze from the southeast, it makes our local weather a little warmer than it would be by geography. And so technically we are in a subtropical region. Advantage and disadvantage.
The tropical dew line does the Tropic of Cancer and Capricorn in between there? Is that tropical?
Exactly. Well, in the middle, tropical is really the equator. You know, up to about 25, is it 25 or 30 degrees north and south of the equator, the whole swath. But the core of the tropical region is the equator, really, which again, the blue line, as you can see, the bright green is mostly around there. Spanning north to about 25 to 30 degree north, south also.
We're actually
Madagascar. We are in a desertic zone, were it not for the sea breeze that brings in humidity from the ocean, picks up all the warm, moist air from the surface and dumps it on the eastern coast. But otherwise, we would be like over here, desertic, the Arabian Peninsula, imagine that. And here is the Sahara and the Sahel, which is the borderline. Okay, a few more details here. Here's some comparison of, these are actual paintings, lithographs of South America and Africa, where we find palms. So it's not just the animal population, where one can say, well, yeah, because animals move, right? By definition, most animals move, and therefore they migrated to that area. Yeah, but how about plants? Plants also migrate, but at a much slower scale, because essentially it's a seed dispersal, right? That will make the plant move, if you will, or the species move as a whole, the population rather, and populate an area. But the fact is that we find palms both in South America and in Africa with a continent in between. And I can tell you for sure, palms do not tolerate salt water as such, okay? Even though coconuts live very near the salt water, they need that thin layer of actual land in which to grow, because the roots do not tolerate the salt water as the mangrove does. And we find what? Lo and behold, we find the fossil record also of palms, all right? What is known actually as the palmate leaf is very definite. This is a palmate almost identical to a palmetto palm of today, all right? And these are fossils that are dated back to millions of years ago. These are also palms from Asia that tend to be smaller in general, but we find on that whole swath, whether it's Africa, Asia, or America, we find palms, right? And so it's also another indication of a common ancestor that spread, and probably they predate the continental drift, so that those populations were there already, or the ancestral population was there in these regions before the actual split. And then once they split, then they diverged into the varieties that we see in those regions today. All right, now I'm going to shift into the molecular evidence, which is more recent, in the past half century, more or less, since the elucidation of the DNA code, and also the establishment, finally, that the molecule of inheritance is, in fact, the DNA and not proteins. This is delicious, by the way. Thank you very much for that. And I say DNA, not protein, because as odd as it may seem for us today, it turns out that in the 30s and the 40s of last century, the best candidate for those inheritance factors that Mendel had alluded to were proteins. Why? Why would you think that proteins were better candidates than the DNA itself?
They are better represented, and they don't degrade.
That's one aspect, but okay, I'll give you some hints. Number one, we knew about proteins, or scientists knew about proteins before they knew about DNA, all right? And the actual sequence of the amino acids, the 20 amino acids occur in nature and so forth, and those were known. What else? There's a great variety of proteins in our body. Our bodies are mostly protein, well, not mostly protein, we're mostly water actually, but there's a large amount of proteins in the body, and a large variety of proteins, all doing very different functions and structurally. All enzymes are proteins, so it was kind of an intuition, kind of a simplistic intuition to consider. And it's a polymer also, a long, very long polymer made up of complexes and so forth. And so it was kind of a natural intuition to think that the proteins were the inheritance factors that Mendel was talking about, until the DNA code was actually cracked, and the molecule was put together by Watson and Crick, right, in the 60s.
But the building blocks of DNA are protein,
No,
correct?
no, they're nucleic acids are different from amino acids. They're rings. Some are simple rings, some are double rings, but they're nitrogenous rings. They're somewhat similar to the amino acids of the proteins. But first of all, there are only 4 instead of 20, right? You have adenine, guanine, cytine, and cytosine, the ATCG combinations, all right? They're organic rings.
Amino acids.
No, those are nucleic acids, okay? Yeah, technically they're called nucleotides. Let me show you a picture that says 1,000 words. Okay. Adenine and cytosine. All right, so here's one, and is that large enough? You can see it over here. These are the nucleotides. Oh, here, this one's better. Okay, here we go. So you see the rings. They're called nitrogenous rings because they have nitrogen in it, but it's mostly carbon, again, organic molecules, carbon rings. Some are single rings like cytosine and thymine. Some are double rings like adenine and guanine. And that's precisely why they have to alternate, so that you have a total of 3 rings, if you will, in the rung. And then that way the rail stays parallel and doesn't bulge in or kinks in or bulges out, all right? At any rate, it's all very mechanical. We'll get more into this next semester or so. I mean, next semester when we look at the beginning of life, because I'll go into DNA in more detail. But these are nucleotides. They're not amino acids. Some are similar to amino acids, but not necessarily, okay? They're different molecules. They are organic molecules. And these are the 4 that form the DNA. These and only these, naturally. And then when DNA is transcribed into RNA, thymine is substituted by uracil, which is almost identical, except instead of a methyl group, it has a simple hydrogen, and there's another thing that changes somewhere. Yeah, there's a single bond here instead of a double bond, all right? But slight little detail makes it called uracil, that's RNA. But bottom line, these are the 4 nucleotides that will be the analogous of the amino acids for the protein in the form of a polymer, a chain. And that is really where the information is stored. This is the genetic information is stored in the sequence of these nucleotides, the nucleotide sequence, and the particular steps that are embedded in that molecule. And that was not evident until Watson and Crick came up with the actual model of the structure. And you've seen those diagrams, that famous photograph of Watson and Crick pointing, let's see, this one. This is a very famous picture, right, photo. This is Crick, who was a PI, the principal investigator. This is young Watson as a grad student in his lab at in the UK at King's College. What is Watson holding, Crick holding in his hand? Notice he's holding, some of us may still remember this gadget. It's a slide rule. Remember the slide rule? For doing calculations, okay? The slide rule, I remember the slide rule in high school. Today there are in museums. This is the precursor to the computer, folks. We've come a long way. But this is the model of the DNA. You see that they're building it with equipment from the chemistry lab, which is used, these bars and clamps are used for holding on flasks and test tubes and so forth. That's what they're using for building their DNA model, okay? Trying to fit the molecules so that you get the pattern that was seen in the X-ray that they stole, that Watson stole from
Rosalind Franklin?
Yes, Rosalind Franklin. Thank you very much, Rosalind
Written
Franklin.
down in the history here.
Yes, indeed. It was a man like because Rosalind was in the other college, right? And well, there are all kinds of. I'm not going to get that diverged too much, but it resist the temptation. There she is. But we'll get into this story deeper when we look at the beginning of life, okay, and get into the into the DNA structure. For now, what we can say is that once it was elucidated in the 60s that DNA was indeed the inheritance molecule, then it all fell into place at the molecular level. And now we can do molecular homologies and look at percentage of compatibility between the genomes of the different species. All right. And we find that there is a compatibility that is greater with animals that look more like us. And when we diverge out to other animals, there is a diminishing compatibility or homology of the DNA or the protein sequence, either one, because they're equivalent, all right? So for example, of course, we're 100% compatible with our own DNA, with our own, okay, so here they're using polypeptides, so they're using protein sequence. But again, keep in mind that we can go backwards on the transcription translation process and we can find the DNA equivalent of that polypeptide sequence. With the rhesus monkey, this one is using, okay, this is using hemoglobin as a particular polypeptide, right? Hemoglobin, which is made up, it's a complex of 4 identical myoglobins, which are polypeptides, and that forms a complex of the hemoglobin. 95% compatible with the rhesus monkey. For example, you've heard of the Rh factor, right? This is where it comes from, the Rh factor. And so we have the 2 most common types of that we use for typing blood is first the ABO group, whether we are A, B, AB, or O. So we know our blood group. Hopefully everybody knows their blood group, and it's somewhere, I think it's in the driver's license or something, right? For accidents, it's very important to know our blood type if we need a transfusion and we are unconscious. So I'm O negative. What is the negative for? Negative is the Rh factor. The nurse can tell us all about the Rh factor, okay?
We're the universal donor.
Exactly, universal donor. But I can only receive from O negative. Otherwise I clot, right? But nature has a way of balancing that out so that the most common blood is O negative.
No, O positive.
O positive is more? Oh, really?
Pretty sure it's O
Oh,
positive.
really?
O negative.
actually,
B negative
wow.
is the rarest form.
It's one of the rarest.
They're
Oh,
the
okay.
They're the universal recipient.
Yes, right.
Let me double check.
Oh, no, you are the
No.
universal donor.
B negative is
Negative, O negative. Which is
O
zero.
negative,
You don't
right,
have
because
RH.
I don't have the
You have
RH,
RH.
right?
And
Yeah,
you
O
don't
negative.
have A and B. So
Exactly.
You are like,
Exactly,
as
blank,
clean as it could be.
tabula rasa.
So you can give it to anyone and it wouldn't change.
Exactly,
Any
there's-
reaction.
Mhm.
The O positive doesn't have A or B,
So it's universal
so
donor.
you can be almost a universal,
And then B
but
negative
you
is the
cannot
most-
give it to a
To
O
a
negative
negative because of the RH factor.
because of that.
Exactly.
You're giving the factor.
Right. So what we're talking about here basically is that the surface of the red blood cell, all right, the surface has many proteins, but 3 of the proteins that it has are just named like that. One protein is named A, the other protein is named B, and then the other protein is named RH, which is what I want to get to. So people who have the A protein only on their red blood cells, those are A type. People who have only B protein, those are B type. The people who have both proteins, A and B, those are the AB. And the people who don't have either A or B proteins on the surface of the RBCs, they're O, meaning 0 actually. The O is a 0. Okay, we have 0, either A or B. So that establishes that blood type or blood group. In addition to that, there's a third protein that is called the RH protein. And all I wanted to say is that it's called RH because it comes from the rhesus monkey.
Oh.
In other words, it's found in the rhesus. And that's why, and probably that's why they're using the rhesus hemoglobin here, is to make that connection. And so the rhesus monkey, because it has 95% compatibility with our RH protein, okay, then it's a good subject for clinical trials before we go to the human, the last step before going to a human trial. So the poor rhesus monkey got it for being the experimental monkey of choice.
The monkey cases and by the zoo.
Yes, exactly. Yeah, they brought also the rhesus monkeys. I understand the rhesus monkeys were the ones in Tarzan, original film, the Mueller Tarzan. They started monkey jungle here, and then they got the monkey jungle. The monkeys got away, and they're all over the place now. So,
Well, we have a story. We have a few monkeys in there.
Macaques, yes.
Or maybe big cypress.
yes,
In
exactly.
certain places they escape
They escape
from some of the lactate that UN used to have it by the zoo.
by zoo. Yes.
And I don't know.
And Monkey Jungle was also around there at some point, and they got wild. So especially after Andrew Hurricane. Mm-hmm.
So
Like,
there's 3 proteins of blood. There's A, there's B.
Yeah. there are many proteins, but we focus on these 3 proteins to determine the types, the blood type.
So A is a protein, B is a protein.
Right.
A and B is the same, it has 2 proteins.
Exactly.
And then O has no proteins.
Correct.
And then
Of
R
A and B,
is a
of
third
R and
protein.
B, of O type has no A and no B.
Mhm.
But in addition to those 2, the third one, RH is independent of A and B. So that you can have O, for example, what is O? Does O have A?
No,
No. Does O have B?
no.
But O can have RH
Oh,
or
I see.
not have RH. So it's an independence.
So then it's either, you can either have 0 protein, oh no.
0, yes.
0 is O negative.
Exactly. That's 0, 0. It's a double 0.
A, B, or O positive.
Yeah. So you have to see it as a double 0. O negative is a double 0. It's a 0 on the AB and it's a 0 on the RH.
Got it.
And that's why we call it positive or negative, because the RH is only one protein, so either you have it or not. And so just positive or negative works for there.
Okay, yeah, I know because my sister had negative and her husband
Negative?
was positive and it was a problem
So it's incompatible.
having babies, yeah.
You're right. Well, yes, the RH factor, right? Because then it sensitizes. The first pregnancy is fine, but by the second pregnancy, she's sensitized to the embryo, to the fetus, right?
What happened in my family, my
Yes.
grandmother, she's O negative. And
Yes.
my grandpa O positive. My uncle, he
Mhm.
barely was born alive.
Yes.
Had that
Was he
yellow-
firstborn or secondborn?
Second. One, she's fine.
Exactly. So the firstborn, I understand, it's fine.
But my uncle is O positive, so
Right.
the RH affected him.
Yeah. And oxygen, they call it, yeah, they call it blue babies because they're born with less, the oxygen, they have difficulty oxygenating the blood. And so they're called blue babies because they need, but now today they do a transfusion right away. And I think they desensitize the mother also somehow.
Yeah, they gave her shots or
Mhm,
something.
exactly. Blockers or antagonistics or something that block the sensitivity. Yeah. Yeah, but those are the blue babies in way back, they actually, some of them died because they didn't know what to do, well, not oxygenating well. Okay, so you can see, but still sticking on the hemoglobin, going down on the animal scale to a mouse, which is not a primate, okay? It's a rodent, is 87% compatibility, the hemoglobin of the mouse with the hemoglobin of the human. And that's why Moose also got it, went into the lab and became also the other animal of choice because of its compatibility at the hemoglobin level and many other proteins, of course. With the chicken, 69%. Maybe a reason why the vaccines were developed in chicken embryos back when, right? The polio vaccine, I think, typically was done on the chicken embryo. All right. With the frog, 54%. They're not even, you know, a bird is not a mammal. Here we are at amphibians, okay? Lamprey, which is a type of fish, actually. We have, oops, sorry. Our hemoglobin and the lamprey hemoglobin has 14% compatibility. Very little, but some of homology, percentage homology.
So do animals have different blood types as we do?
Yes,
They do.
they do, okay? They may have more or fewer factors than we do, but each one will have, because the surface, really the surface of every cell is very complex. It has all kinds of molecules there, and those molecules can be antigens, can react either with foreign bodies or with antibodies, okay? And that's what the antibiotic is, precisely, it's detecting the surface.
Some of them green, I mean, I think that.
Yes, when you get down to the amphibians and the fish, they have a greenish blood, the amphibians in particular, or sometimes bluish, all right, depending on the species.
Is that if their hemoglobin is based on something other
Right, it has to do with the reflection, yes, exactly. And the center, I think that for amphibians, they said they have a nucleus of copper instead of iron. I think it's copper. Yeah, the nucleus is, that's what makes our blood reddish, is that the iron oxidizes. So iron oxide, another name for iron oxide is rust. And what is the color of rust? It's a reddish color. So oxygenated blood is more reddish than venous blood that is non-oxygenated and got CO2 instead of oxygen. Okay, when we expand this issue of percentage homology, we can look at some genes that are regulatory genes, are known as regulatory genes. These are genes that kick in at different stages of development, typically in embryonic development, for developing body parts and entire body plans, like the whole dorsal ventral sequencing and the sequence of events of when these body parts are to express, all right? So for example, we want the internal organs to express before we start getting the external organs that are going to be on the outside of the body. And those are known as regulatory genes, and they kick in, they activate at different stages during embryonic development. So because they are regulatory genes, they have a fairly large percentage homology. And those regulatory genes are key candidates for matching with different species and seeing the percentage homology between different species to see how related they are or not. So for example, here we see, what is this? The Hox gene, yes. The Hox gene is called Hox because it determines what is known as the homeotic box. The homeotic box is kind of the core, the trunk of the body, and what happens there, and the segmentation of the body, which follows the general body plan for animals as head, trunk, and abdomen, all right? Head, trunk, and abdomen is a general body plan for animals, and that is known as the homeotic box. And because the genes that regulate where the head is going to occur and what happens inside the head and where the trunk or the middle section, the thorax, is going to occur, and then the abdomen and its organs, they're all in this region called the homeotic box, or Hox for short, all right? And so these are the Hox genes. And what is shown here is the percentage compatibility of Hox genes between different animals that are distantly related. For example, this is a roundworm, a nematode of some sort. This is a beetle, so this is obviously an arthropod, right? A Coleoptera arthropod, invertebrate with an exoskeleton. This is a nautilus. So this is a mollusk related to the octopods and squids. A starfish, which is an echinoderm, they have a skin outside, so they have technically have an internal skeleton, and they have a layer of skin on the outside, including the sea urchin and the sea cucumbers and the lilies, the sea lilies that we saw earlier, the fossil. This one is a amphiox, which is a primitive type of fish, all right, with gills. And you can see that it has some percentage of compatibility. The ones that have an internal skeleton, like the starfish and the lancelet, they have more percentage compatibility than with, say, the roundworm, the nematode. And so we can establish these. This is essentially a cladogram, but it's a much more sophisticated cladogram because now it's not based on external morphology, all right, because a starfish and a fish don't really look alike at all on the external morphology. But when we start comparing the percentage homology of genes or proteins, we find a greater compatibility there. So they will be put in the cladogram closer to each other than other animals that perhaps look more similar on the external morphology, but are not as related based on the DNA sequence or the genome sequence. So this is really the latest craze on forming these cladograms, or the tree of life, looking at percentage homology, either on the protein sequence, which is easier to obtain, or if we want more detail, we get into the actual DNA. But the DNA then requires sequencing the genome of that species, which is more elaborate, more expensive, but it's being done now. The big one was the human genome, right, in the 1990s, the project, the Human Genome Project. That took about 10 years. And then to refine it down to what it is today, it took another 2 years, more or less, to 2002, more or less. Now, sequencing is becoming a little less expensive and a little faster. More and more animal and plants genomes are being sequenced, right?
Father, last class when I asked, we briefly mentioned like the elephant, because you were talking about symbolic language.
Yes.
And understanding depth and how much we know they understand, but obviously elephants. This week I was watching something about octopus, and they were talking how they actually can conceive of a concept of, I don't want to say before and after, but planning, that they can move their shell. So at some point, will the genetic information be supplanted if we understand there's a higher functioning? Because, or do you just say at some point the mollusk class goes up as high as the octopus and that's it? Or would the octopus, because we understand function, that could be moved into a different.
Right, so when we think in these terms, the operative word here is branching, okay? And where we put the branches, where we put the nodes of the branches. So what could happen is with further information, those nodes may change somewhat, all right? But they will still stay within the major groups, because at this point we've established the major groups, and I'll show you the diagram. Go back to where we started 2 classes ago with the sophisticated diagram of the tree of life. But also this gives me an opportunity to mention, because you mentioned behavior essentially of the octopus remembering certain things and doing some things before and after and so forth. So having some concept of time, maybe, right? Okay, but we would include that in what is known as behavior. And there's a whole branch of behavioral biology that has developed today from anywhere from, remember Conrad Lorenz? I don't know if Lorenz rings a bell, but he did the experiment with imprinting, and he would have these little ducklings following him because they thought that he was the mother duck, right? Because that's the first thing that the ducklings saw when they came out of the shell, all right? It's called imprinting, and many animals have it. They fix on whatever first animal they encounter, thinking that it's their mother, and they'll follow that. So that was Lorenz with the early experiments on animal behavior. And since then, It's a whole field, a whole career of behavioral biology. What I'm trying to say about all this is that behavior is also part of the phenotype. It's an expression. It's part of the phenotype. It's not a physical thing. It's not a physiological thing, but it has a physiological substratum. The behavior has to happen somewhere organically. And where does it happen? It happens on the brain. Mm-hmm.
Are you going to say that's an absolute, or is that up for?
No, what I'm saying is that behavior is phenotype. Biologically, behavior is phenotype in contrast to genotype.
This is what I'm asking now.
Right.
Are there scientists or some people, because the name that's coming in, I don't know enough about his work, so I know, like, I'm thinking Stephen Pincus out of Yale and some of his, but is the idea of what you're saying that behavior, it's given, it's an absolute, or are there people who think that it could be?
Oh, you mean deterministic behavior as opposed to free will?
I haven't gotten there yet.
Yeah. Well, that's the discussion right now. Okay, but probably I remember my PI at Arnie Stein at when I was doing the genetics at Purdue. He said we probably, and he was a genetics expert. Says we're probably more determined than we would like to admit in our behavior. Okay, we're probably more determined than we'd like to admit, even though I truly believe in free will, and it's part of our teaching, it's part of our doctrine, and I am convinced of free will. How can we will evil? Otherwise, we wouldn't logically choose evil if it was just 100% determined behavior, because evil, one way or another, comes either against us or against others. But anyway, he was saying we're probably more determined that we like to admit. It's a humbling thing because we like to think that we are that we have total or absolute free will, but we don't really. Okay, a lot of it is determined at a very sophisticated level, for example, of hormones. But we know that hormones can be very, very, very strong, and they can be very strong on the young as they're growing and so forth. But still, we maintain that free will can still override the hormonal influence and still allow a person to behave ethically. All right, so anyway, all I wanted to say about that is that behavior biologically is also considered part of the phenotype.
This is slightly off topic.
Okay.
Because the
Go right ahead.
part brings it about it. I would assume it only affects students, but where does talent come into all of this inheritance thing? I mean, my grandmother could paint beautiful pictures. My mother couldn't. I can't. Is there a hereditary factor, or do we know?
So talent, or the expression of talent, right? Because people may have talent and they either don't know it or they don't exercise it. But let's talk about the expression of talent, for example, in painting. And it's very obvious sometimes when a person has a painting talent that we can see it, right? It's an extremely complex behavior, I would say, and so many factors go into that expression of talent, including the mood that the artist feels in, and I don't know, the temperature or the environment that they're surrounded by, and what is influencing or not. It is said that Michelangelo was inspired when he did the ceiling of the Sistine Chapel by the clouds, by the figure of clouds, and I think that we've all at some point in our lives seen figures in clouds, either people or dragons or animals and things floating around, right? And so it's an extremely complex behavior, the expression of talent. And therefore, by complex behavior, it means that many, many factors fit into it or feed into it. And therefore, for one talented person, it may express in a particular way. For another talented person, it may express in a different way or not express at all.
But something like singing.
Yeah.
If you were capable of.
Right.
I know
Now.
nothing about music, but the different ranges that
Yeah.
some people can sing would seem to be also genetic.
Absolutely. Not only that, perfect pitch.
Oops.
Talk about singing.
Very
Perfect pitch. My sister-in-law, she has perfect pitch, and she's a soprano. She's operatic soprano and whatnot. It's amazing. She'll hit the note right where it is. And when she hears music that is dissonant, it's very distressing to her because it's hurting her ears. to hear music that's dissonant because she just has perfect pitch, which means we can take it down organically to the way those little bones are, the stipules and all the little bones in the inner ear are detecting that sound that are just the perfect design for getting that pitch just right, okay? So that definitely has a genotypic background to it, no? We're talking about some organic thing in the body, right? And then not only that, but it's also a confluence between not only the good ear, but the good throat, where she has control of the vocal cords, so she can mimic, she can reproduce that perfect pitch, because she could have the ear, but not the vocal cords to do it, to reproduce the sound. So extremely complex behavior, and is there interaction between the phenotype and the genotype? You better believe it, in a very, very sophisticated way, all right?
We can't.
Well,
No.
you can mention capacity to do things.
Right.
And very, very, very complex things. Now, but then you have to jump to the issue of how do we acquire that?
Right.
Or how do we use it? And that goes into a philosophical realm.
Right.
Because you can explain the mechanism.
Right,
Because
how?
maybe even miracles have some sort of explanation at a physical level. They might
Physical,
be
yeah.
extreme, but they happen. That's why you can visually or sense them. But that doesn't mean that they're going to happen. There's a capacity, but how they happen, that's a combination that goes beyond the natural the statistics.
Exactly. It does. It goes into the metaphysical. You brought up two interesting points. You talk about capacity, which in science we talk about capacity. The equivalent of that in philosophy, we talk about potential. Okay. For example, if we are American citizens, we all have potential for being president, but we're not actual presidents, but we have potential. That's philosophical. Mm-hmm. And whether that potential actualizes or not depends on many, many factors. And the other thing about the, you were saying the miracles, yeah, which even medically there's a description for it, but it's just to kind of phrase it, to give it a name. It's called spontaneous reversal, because I've seen this in the cause of beatification or canonization for saints, right? They have to have that miracle, and on the person who is healed physically, physiologically, there has been a spontaneous reversal of a necrotic process, of a process of illness. Spontaneous reversal, like say, from a cancer cell, that not only does the cancer stop, but it shrinks. Mm-hmm. Yeah, exactly. You can go deeper down into the molecular and so forth. And if there's something physiological, there will be an explanation for it, but the explanation is still the how. It doesn't say anything about the why. Okay. And so that's where science has its limits. Mm-hmm. When philosophy kicks in. Or theology, which is really applying philosophy to the metaphysical with a belief in God. Okay, so I just have a couple more slides to show you here to prove the issue of I was talking about the Hox gene earlier, right? How this box of genes, this toolbox, is the one that determines the head region, the thorax region, and the abdomen region, so that it happens in this sequence, excuse me, and no other, so the thorax is not in front of the head and vice versa, and so forth. And the percentage homology you can see here on the Hox box, on the Hox genes, from the fly. This is the fruit fly, right, Drosophila, that is also an animal of choice in the lab because you can get a generation very fast. And the human, at the embryonic stage, it has the same pattern, the head pattern, the thorax region, and then the rump or the abdominal region, okay? And those genes are in the percentage of homology here, indicated by the different colors. This is based on the actual genetic sequence of the genes. So percentage homology is occurring across the board. When we start doing these pictures, it starts getting mathematical. These are the diagrams. This is the sequence, the DNA sequence, GGAT, This is actually the rungs of the ladder, just one side, because since they're complementary, as long as we have one side, we can interpret the complement. And so we only need one side of the ladder. Guanine, guanine, adenine, thymine, guanine, cytosine, etc. Right? Okay. This is a sequence. They didn't tell me of what they're sequencing here, but you can do these now because we have the genome. We have the human genome, basically the 3 billion base pairs that determine the human genome, right? And we have the genome of the lemur, the chimp, oh, that was a bird up here. And the dog, the cat, the cow, and the pig. We have all these genomes. Now we can do blast. It's called a blast, a genome blast. We take, let's say we start with the human genome. We take this sequence here of 50 nucleotides and we blast it into the chimp genome. And this is a percentage homology that we get, all right? Which is, let's see, there's one difference here. Another one, 2. Tell me if I miss any between the human and the chimp. 3. That's it. In 50, only 3 differences, all right? So in 106 differences on this particular genome. So we can say that 94% just based on this particular sequence, 94%. In fact, when we do a whole genome blast, we get about a 99% compatibility with the chip. And so on, the same with the lemur. So we compare human to lemur, and we're going to see that there are going to be more differences. Here's one, the T instead of GG, right? So that's one more. Here's an A instead of a G and a C, etc. So we have less compatibility with the lemur, but we have more compatibility with the lemur than with the bird. I don't know what bird they used here, but they used a bird, okay? Probably the chicken. And so we established this percentage homology. We can also go to these other animals and do the same and get this percentage homology. So we see that the human and the dog are much less related. So we have to put them out in a branch that is further out here beyond the lemur. And then we can also compare dog and cat that are very similar. Okay, let's see. So far, identical. Here's one, C, T. In this sequence, there's only one dissimilarity between the dog and the cat in this particular sequence, which involves about 38 nucleotides, all right? So if you extrapolate that to 100 to make a percentage, it's about 3. According to the sequence, only 3% difference between the dog and the cat genome. So they would be closely related, both carnivores, and so on and so forth. So you seek in all these matches by doing a comparative percentage homology between different species. You come up with essentially a cladogram of their percentage homology, or percentage compatibility. This is showing essentially the same, the homology, percentage homology, but in different groups of animals. So it's comparative homology between different groups of animals. It turns out that just like there is a half-life for the isotopes, isotope radioactive decay, where we can get an actual date out of it, okay, a chronology. Also molecules, because of the background rate of mutations. There's a standard mutation background rate that occurs in nature, okay? And using that background rate of mutation, and looking at specific mutations that occur between different animals, we can establish a molecular clock of divergence, estimating when it was those animals or those organisms diverged, those two species diverged, based on the number of mutations that they have compared to the background rate. So here is on the plot, see if I can make it a little bigger, more centered. All right. So we have rate of evolution of DNA in constant. This is what I'm talking about, the background rate, okay? It's a constant rate of mutations. The scale is in millions of years, so it's a very handy scale. And we compare this rate to the rate of mutation between two specific species, and we can see where they fall on the chart, what kind of percentage substitutions they have of nucleotide, single nucleotide substitution, which is one particular type of mutation, and just extrapolate down to how many millions of years that mutation should have occurred. So it gives us an approximation of the divergence, the time of divergence, where the node would be. Okay, so all this to say that we can also use the rate of mutation, the background rate of mutation in DNA to establish a molecular clock. And this one, the scale is typically in the millions of years, and that's how we can determine the branching or estimate the branching of different species. Always happens to me with these little bars. Okay, so here's a sequence, an original sequence. After 25 million years, there was a mutation here, GT, and another one here, TG, which is estimated according to that table to be about 25 million years ago. A second mutation here, also from this T to that G, to that C, cytosine, and here a C to a T. Okay. So we can see that these two sequences are getting further and further apart from each other because of the individual independent mutations that are occurring in each one of these sequences.
Are those more animals or plants, or where do they get in the DNA? To make it
Any DNA sequence, any DNA
No,
sequence.
I know, but if you go to the fossil record,
Right,
you won't be able to get any DNA from that.
correct.
How they're getting 25 million years.
This just a style, oh, I see what you mean. Yeah, and the scale here, right. So this is an extrapolation of these dates. Let me see. It turns out they're actually using proteins here because they're extrapolating from the protein, the DNA sequence. And that's how the scale is established. Of course, this is a slope of the line, right? The experimental dots are where they're falling. So this is a slope, and that's why it's an approximation. But it looks like they're looking at protein sequence. And from the protein sequence, they are elucidating what the DNA would have been.
So that's a model, basically, more something that they come out with.
It's a what?
It's a mathematical model.
It's a model. It's a model, yes. Yeah, I need to find a reference for this to see what kind of samples they're using. Yes, yeah, it's a mathematical model, but it's based on the mutation rate that is known today as background, which is 1 in every 10,000 bases. There's a mutation rate. Okay. And again. So based on that molecular clock, different animals can be placed into a scale with the average percentage of DNA change, that this is equivalent to the mutation rate. The non-coding DNA, which in the human at least is about 95%, 97% of the DNA, it also has a sequence, so it's informative in that way. And by looking, of course, these are all modern animals, and it can be established the percentage compatibility between them based either on the DNA, the non-coding DNA, the protein DNA, meaning the coding, right, the other 3%, and also looking at ribosomal RNA. Our RNA is ribosomal RNA, the sequence in that RNA with percentage homology or compatibility. If we extrapolate from the line, it gives us the millions of years where diversion would have occurred. And again, what is interesting about this is that the animals and plants fall where the expectation is. The ones that have more common characteristics, for example, a vertebra, then they fall closer on the line. The ones that are further away from us, morphologically, are also fall in less percentage compatibility. For example, down here, yeast, which are actually nucleated, they're eukaryotes, but they're single cell colonial. The end result of all of this is this map that I showed you at the beginning, which is a more sophisticated map and more realistic branching of the species based on molecular compatibility. So this goes beyond the gross morphology that we did with the 5 standard kingdoms, right? This is a much more sophisticated map. And this one is based on molecular homology or percentage homology, which the animals are over here. Just to get an idea, for example, most of these are microscopic. For example, amoebas are here, radiolarians, foraminiferans, these are all what we would normally call animals, microscopic animals. They used to be put in that protozoan category, which was the grab bag, the protozoans, right? But that's when I was in high school biology back in the 60s. Mm-hmm. Plants over here. Alveolates, they also, if we would look at these animals on the microscope today, we would normally intuitively place them as animals, as what we call animals, simply because they're moving and they're heterotrophs. In other words, they don't produce, they don't photosynthesize, they feed on other organisms, right? But that really would be a very primitive characteristic. Their DNA is so different from the animal DNA that they're being placed in their own branch here. Mm-hmm. And also these groups, heteroconts. See if I recognize any of these names. No. These are mostly microscopic organisms, mostly in the ocean. The molds are here, that's interesting. The slime molds, which we normally classify as mushrooms, right, as fungi. And this is for the ones, for all the cells that have a true nucleus. The ones that don't have a true nucleus, either the bacteria or the archaea, you can see that they have quite a branching pattern also. And then this root, which is anybody's guess, but that would be what is known as the first common ancestor. I have to go back about 4 billion years. So that is really the sophisticated map that is coming out today by doing the percentage homology. The one that I showed you was a simpler one and the one that most people speak to, even to this day. Even though this one can be broken up really, the algae are photosynthesizing, so they're technically in the plant kingdom. And Moneras include both bacteria and archaea. Then all of these others, this is looks like a foraminiferan, the euglena, the paramecium, they would be put in different branches here, somewhere in the eukaryotes, because they all have a true nucleus.
The euglena is a very interesting organism. Because
Yes.
It has photosynthesis, but also does heterotrophy.
Yes, it also moves and eats and can eat by phagocytosis, right?
I was in school.
Yes.
There was talks that it could be a precursor, plants or some kind of missing link.
Right.
But now I see that they put it farther away from plants.
Yes. Mm-hmm. Yes. Again, we have to avoid the trap of saying that any contemporary species is a precursor, right? Because it's a contradiction in term. If it's a precursor, it by definition doesn't exist now as living, all right? Now, there are some that we call living fossils, but typically they are dead ends. In other words, they are a species on their own, like the coelacanth, that huge fish that lives in the deep ocean. You've heard of coelacanths? They're called living fossils simply because they're so primitive, and their fins are lobed fins, and they actually do use the fins somewhat to move around, especially the lateral fins, the pectoral fin, and the anal fin to move around on the bottom, caudal fin. But they are fish, okay? They're bony fish, and they have retained a very primitive looking structure, which may put them back, I don't know how many millions of years, but they're fish. So they're not an ancestor to anything because they're living today, okay? And that's where perhaps some of the disbelief, if you will, of fundamentalism with regards to evolution is that they think that we're talking about, for example, in the case of the human specifically, deriving from the monkey, but we can't because the monkey is also living, we're contemporary. We can't have contemporary ancestors, all right? That's anachronistic, and therefore they see an insult in that, right? In that if we're created in God's image and the monkey is not, for sure, then how can we derive from the monkey, all right? So when we understand the concept that there cannot be any contemporary ancestors, then perhaps it's a little more palatable for these people to appreciate the process of evolution. Now, having said that, we can also talk about living fossils in a very loose term of the word, simply meaning that these are some organisms are very primitive. It just means that in the branching and the cladogram, you know, they have, we can make that argument, for example, for yeast, which branched off thousands of millions of years ago, and they're still around, okay? So we can say, well, maybe yeast is a living fossil in the sense that it branched off early and it's still around, at least one variety of it, or several, I don't know how many species of yeast there is, but they branched off early on, and that makes them more primitive than the other animals that we see around. But they're not, the one that yeast that is living today that is used for rising bread is not an ancestor to anything because they're living today. All right, so just to wrap it up then, what we have is evidence from these 6 areas that come from different parts, actually. One is geological, the other one is gross morphology, the other one is the embryonic development. And you can see that they're really independent of each other, and they all point to the same mechanism, especially when we add in the molecular evidence today of the percentage homology, either for proteins or for DNA or RNA. They all point to this branching effect of common ancestors, right? Now, what we're going to do in the next few classes is we're going to go into the mechanism of evolution. How can this happen, actually? How can this happen in time on Earth, on the surface of the Earth, where we have life, whether it's on the land or the oceans, the mechanisms. And it's going to come down to these 4 forces or 4 factors. Meyer has more, but they can actually be grouped. I think he has about 7, but they can be clustered or grouped into mutation, migration, gene flow, and genetic drift, okay? And those are the ones we're going to get into and see how is that possible, how can that work in nature, to see at a possible mechanism how this could work. Ultimately, it's going to be selection, natural selection, that is the driver for the possibility of evolution, of a new species emerging. All right, questions, comments? No?
Should we be reading further in there?
Yes, so chapters 2 and 3 of Meyer, okay? Chapters 2 and 3, and you'll see how you'll find I've tried to merge the lectures with Meyer's reading or chapters, and then supplement it with some other stuff. Okay, now next week, let me just check the calendar for a moment, because I know that one Saturday I'm going to be away, but it's not coming up yet. Is the 19th, is it?
I think it's
No, actually, let me see.
the week after, you're there the 19th, 11/2 is the next class. So 9 and 7 is what, 26?
Yes, the 26th, I'm going to be away. 26th, yes. So let's see. Next Saturday, we still have class. And the Saturday after that, October 19th. So the 12th and the 19th still have class. And then on the 26th, I'm going to be away. And then we come back on November 2nd. All right? So, okay. Thank you very much. To be continued. All right, my pleasure. I'm going to stop the recording now. Okay, Ada, stopping the recording.